Hello, and thank you for joining today's webinar at the New York Botanical Garden. I'm Lawrence Kelly, Vice President for Science Administration. It's my pleasure to welcome you for today's presentation, Novel Tools to Understand Biodiversity. At any time during the webinar, please submit questions by clicking the Q&A button at the bottom of the window, and we'll gather these questions for our speaker to answer at the end. Live captions can be enabled by clicking the CC button, then show subtitles or view full transcript. Today's presentation is being recorded and will be available in the NYBG lecture library. You're joining us for a special seminar from Dr. Damon Little, who's up for promotion from Associate Curator to Full Curator. In addition to his research on the systematics of conifers, Dr. Little creates tools to help botanists work more efficiently to catalog and protect biodiversity. The development of these tools and Dr. Little's expertise starts with fieldwork, herbarium specimens, and DNA sequencing, with modern applications involving remote sensing, artificial intelligence, and machine learning. Underlying this work is the inextricable connection between plant diversity and human survival, and the need for all of us to work quickly to understand and conserve plant diversity. To introduce today's speaker, I'm happy to turn it over to Dr. Greg Plunkett, Director of the Cullman Program for Molecular Systematics at the New York Botanical Garden. I turn it over to you, Greg.
Thanks, Larry. It's a really great pleasure to be here this morning and to introduce our speaker, Dr. Damon Little. Damon first joined the staff at the New York Botanical Garden in 2005 as a postdoctoral researcher. And after having completed his bachelor's degree at the University of Vermont and his doctoral studies at Cornell University, he was appointed to the curatorial staff at NYBG in 2007 as an assistant and later associate curator of bioinformatics in the Garden's Cullman Program for Molecular Systematics. And as Larry just mentioned, he's currently being considered for promotion to the rank of full curator. While at NYBG, Damon's combined his talents in the field, lab, and herbarium with his amazing skills in programming and all things information technology. We know a few, if any, scientists who combine Damon's mastery of both botany and IT. And he's been very generous in using these talents to the great benefit of his colleagues and students here at the Garden and to the broader scientific and educational communities at large. And now Damon is working on a very impressive set of projects to incorporate artificial intelligence into approaches to use biodiversity and bioinformatic data in new and creative ways. I'm sure we'll hear about some of Damon's many accomplishments during this presentation, but given that he only has 45 minutes to speak, we can be sure that it'll only be the very small tip of a very large iceberg. With that, I'll turn it over to Damon for his presentation entitled Novel Tools to Better Understand Biodiversity. Damon?
Thank you very much, Greg. Get my screen sharing going here. Good morning. So as Greg said, I will be talking about novel tools to help us better understand biodiversity. Before I actually get into my talk, I want to show some acknowledgements. It was really difficult making this slide in the sense that I collaborate with a large number of people, both at NYBG and neighboring institutions and internationally. And fitting 161 names on a single slide was a bit of a challenge. I ended up using an algorithm to do this, which randomly assigned the colors and the fonts and the positions to everyone. However, the sizes I defined by the number of emails we've exchanged in recent years about scientific topics. I tried to filter out all the kind of administrative ones. And so here you are. If your name is a little smaller than you were expecting, you have to send more emails. If it's a little bit bigger, maybe you send too many. Before I really get into the meat of the talk, I want to kind of define and distinguish between artificial intelligence and machine learning. Artificial intelligence is a much broader term. The key aspect of artificial intelligence is that programmers don't define the algorithms that are actually used. The algorithms are sort of generated from other algorithms. And this is important because we as programmers can anticipate a lot of things and can make quite clever algorithms, but we can't cover all instances, particularly when things are difficult to distinguish among. The hallmark of artificial intelligence is that predictions of some kind are made by data. And the sort of holy grail is unsupervised learning. This is essentially what children do as they learn to speak by acquiring language, learn to read. They're basically given pieces of information and they assimilate them on their own. Artificial intelligence per se doesn't exist yet. It's more the dream, the aspiration to make machines more like humans. And of course, a lot of the things we would like artificial intelligence to do, such as synthetic thinking, cross-apply knowledge from different domains, have creativity, and in some ways have emotions. Of course, we don't want all emotions. Who wants a petulant digital assistant, for instance? And since AI does not actually exist, we all have to think about it, how it's treated in our imagination, particularly in science fiction, where it unfortunately usually ends badly. I suppose this is because if it ended well, it would be a boring story. If you don't want to hear spoilers for a 50-year-old movie, please tune out for a second until I change to the next slide. But I want to talk about 2001: A Space Odyssey briefly, because arguably the star of this movie is an artificial intelligence, HAL 9000. This is, in many ways, the embodiment of what would be the nightmare of a smart home. HAL is the ship's computer, and he controls all aspects of the ship, everything from turning on the lights to setting the course in the propulsion system and maintaining life support. But he also interacts and entertains the crew, and kind of keeps them company. Unfortunately, HAL was trained in a strange way. He was trained once to never lie, to always tell the truth to the crew members, and that his job above all else is to protect them. Someone decided that this mission that he's about to go on is top secret, and therefore they can't tell the crew what the mission is. And this cognitive dissonance leads HAL to have what can only be described as a psychotic break and try to murder the entire crew. That's not what people want in AI, but it is an obvious sort of implication that AI will behave strangely when given crossed inputs. So machine learning is essentially a narrower term that describes the current AI technology, the things that we can do that are sort of aspiring to be AI, but are not yet an artificial intelligence. And again, it makes predictions based on data. And there is such a thing as unsupervised learning, where data are basically shoveled into an algorithm that then learns from it. This is not yet as sophisticated or as successful as supervised learning, which data is also shoveled into an algorithm, but in this case, it's very structured data. And the structured data makes it possible for the machine learning algorithm to learn and generate predictions. There are a whole variety of techniques within machine learning, everything from kind of simple regression sorts of things to the familiar artificial neural networks. One thing about machine learning that's very important is that it's incredibly effective for pattern recognition sorts of tasks, which it turns out most of botany actually is a pattern recognition task. Unfortunately, AI falls victim, or machine learning falls victim to the Dunning-Kruger effect, which is to say that it greatly overestimates its knowledge and abilities given particular input. This can be seen anywhere from self-driving cars going on railroad tracks instead of roads, or making wrong turns down one-way streets, to misidentification of plant specimens. I want to give a brief history of machine learning as it relates to botany in particular. And so the place to start is a classic data set called the IRIS data set. Now, this data set was put together by Edgar Anderson to solve a taxonomic problem. It is not at all a machine learning data set, but it was quickly adopted by people interested in early machine learning. And in fact, the data set itself, which is of 3 species and 4 characteristics of each one, was published before the main book that bears the data. And Fisher wanted to apply his algorithm to solving a taxonomic problem, which is how do you tell these 3 species apart given those measurements? And he was relatively successful in this. There were other similar attempts at kind of multivariate diagnostic tools, but these are kind of what we would term simpler machine learning these days. The first hint of an artificial neural network, the definite progenitor of what we use today, is the perceptron. And that is from 1961 and later refined in 1963. It is capable of quite a lot of things, but is not fully capable because essentially it needs more computer power than was available at the time. And really the next breakthrough that I want to talk about is from 2012, which is a ImageNet data set. This is an image classification data set, and in 2012 it became possible to actually analyze this data set in a meaningful way and get meaningful predictions. This particular innovation in convolutional neural networks is something that we've built upon a lot in recent years. And I want to have one last highlight, which is a bit difficult to understand if you're not into neural networks, but essentially as data flows through the neural network, the residual or skip connection allows the data to be split and processed in 2 or more different ways and then recombined together. And that actually allows us to make both bigger networks and much more accurate networks that can perceive different kinds of detail in the same image, so both fine-grained and coarse-grained details. So as a general overview of my talk, I'm going to talk about biodiversity inventories, followed by biodiversity informatics, a bit on diagnostics, and some on phylogenomics towards the end. Biodiversity inventories are a wonderful thing. This is in many ways the reason why I went into botany in the first place, to go on grand expeditions into unexplored areas. And this particular project is not my own, it's actually Kate Armstrong's. But I really enjoy helping her with it. Northern Myanmar, where she is doing her diversity inventory, is very rugged, as you can see from this photograph. There are a lot of mountains and valleys and rivers, a lot of microclimates and varied geology and varied floristic influences, meaning there's an incredible amount of diversity in a very narrow area. It is wonderfully beautiful, but also very difficult to traverse. As a result, there are a lot of bridges, some of which have slightly questionable design, many of which have very questionable construction, and a lot of which are incredibly scary. Just so you know, that's about a 200-foot drop from the bridge down to the water there. But it's all worth it because after crossing all those bridges, there are lovely conifers at the other side. A biodiversity inventory is basically founded on the expedition. Many other disciplines have in their word root something about study. Botany does not. It's an ancient Greek word that I'm not going to pronounce because I'll do it incorrectly. But my favorite translation of it is to go forth and gather fodder. So we go on an expedition. This is Punganrazi at 4,000 meters near where that conifer picture I just showed you was taken. And importantly, we collect, dry, sort, and pack the specimens in the field. This is a huge task, and in the trips I've been with Kate, we've collected more than 16,000 specimens that have been distributed, both within Myanmar and to other herbaria, most notably Royal Botanic Gardens, Edinburgh, and of course, our own. All those specimens have to be identified, and if there are new species, they have to be described, and this is one of the ones that Kate recently described, a Hoya from northern Myanmar. Once we know what all those specimens are, we have to go to the herbarium and compare them to every other specimen that we can find in that area, as well as the literature, to make a grand list of all the specimens, and eventually flesh that out with morphological descriptions, geographic maps, things like that. And I'll be talking about the processes from identification through dissemination as I go through this talk and ways that we can improve them. Now with the pandemic and coup in Myanmar, we can't exactly do field work there. And so it got me thinking about other ways that we could do some of the things that we need to do. And that's where this project Vertex, which is just kind of in its infancy, started. And the idea of Vertex is to calculate the alpha and beta plant diversity from satellite images. Now, you might think that's not quite the same thing as an actual inventory. It's not a real list of species, and you're right, but it's a very powerful tool. If you imagine the world divided up into grid cells, you can imagine the biodiversity in some cells is much greater than it is in others. The ability to know which areas have greater biodiversity versus others is a wonderful tool, especially if you can think about it changing. So this is that same volcano after it exploded a few months later, and of course, ash everywhere, and the biodiversity is now 0 in all of those spots. And you probably could hear about this on the news or something. You don't necessarily need a satellite image to tell you this. But if you imagine more subtle things like a landslide or a drought, disease spreading through a forest, or even just more utilization than typical, and being able to sense that remotely, not only to plan and implement conservation policies, but also to plan field work and know which areas are worth protecting and which are not. So just to kind of zoom in on a cell, basically what we'll be doing with our neural network is calculating the number of individual woody plants in each cell and the number of species. And if we do that for neighboring cells and calculate the difference between the number of shared species between cells, we can calculate all types of beta diversity. There are many different versions of it, but they use those variables in different ways. So if we calculate the variables, we will have the answer. I want to show you a little bit about the satellite images we're actually using. So this is a picture from Google Maps of part of Finland, which I've become fond of, not because I've been there or have any personal connection with it, but because it's a good kind of test spot in terms of testing out algorithms and various permutations. So right here in the center is a point, which is the center of the images I'll be showing you. And this is from Google Maps. They're a little, we'll just say, cagey about exactly what data they're showing you at any given moment, but I believe this to be a Landsat 8 photograph. We're actually planning to use Sentinel data. So Sentinel is from the European Space Agency, and satellites see in parts of the spectrum we don't normally see. So the first one they see, this particular one sees in, is ultra blue, which is used to detect basically water and the water vapor in the air. And then it has your typical red, green, and blue bands, and these are at 10-meter resolution. And this is part of the reason why we want to use Sentinel data. The Landsat data is at 15 for one band and then 30 or worse for the rest, whereas the Sentinel starts at the low end at 10 meters, which is obviously much higher resolution. There are 3 bands that are considered red edge, and there are 3 different parts of the spectrum. And if you haven't memorized your chlorophyll absorption spectra, you may wonder why those. Well, the answer is that it's right at the tip of when chlorophyll stops absorbing. And so this is when you see all of the other structure, internal structure of the plant, other pigments kind of come to life, and that allows you to better distinguish between different kinds of vegetation. There's also 2 near infrared bands, one of which is at much higher resolution, but narrower wavelength or narrower band than the other. And so in combination, they give you quite a lot of detail. And because infrared is good at cutting through certain types of clouds, you can often see something in the infrared image when you might not see something in, say, the blue, green, and red images. And lastly, there are 3 bands that are essentially used for detecting ice, snow, and clouds. And these can be used by the neural network to know that it shouldn't be paying attention, essentially, to the other bands that are being blocked by any of these other things. In addition to the Sentinel-2 data, we would like to use Sentinel-1. And Sentinel-1 is a synthetic aperture radar satellite. Synthetic aperture radar produces images that are a little difficult for a human to interpret. It kind of looks like noise. But if you look carefully, you can see some of the topography of the area. But most importantly, what you see is the density of the canopy and the structural complexity of these trees. These are, of course, a northern coniferous forest, so there's not a whole lot of structure, but in a tropical forest, this is quite indicative of how many canopy layers there are and what kind of trees there are. So when this project gets going, what we're doing is we're comparing the satellite data combined with forest plot data, so exhaustive inventories of the forest plots. And with that, we can train a neural network to learn what the diversity is from the satellite image. Once we have this, we need to conduct some ground truth validation. So go out into the field and collect from particular pixels that the neural network is predicted from, and then we can see how well or badly the neural network is working, and then fine tune it based on that data. And lastly, I would like to optimize the neural network so that it works in a web browser, kind of like a Google Maps extension, so you can look at a particular part of the world and see biodiversity information at your fingertips. This will have great application for remote monitoring, conservation assessments in a scale that we can't possibly do now. And in the future, when we're planning field work, it will tell us whether we should go up the mountain on the left or the right, which one has the greater diversity, which one has diversity different from what we've already collected from. One way that I would like to do some of the ground truthing is by airborne eDNA. And at the moment, in collaboration with Allison Granberry and high school students from the Hostas Lincoln Academy of Science, which is a public school in the South Bronx. It's a bit under-resourced, but the students are amazing there. And so we have taken some students under mentorship. There are 4 of them this year, and they are working on an urban barcode project submission that is a science fair run by Cold Spring Harbor Laboratories DNA Learning Center every May. And so they're going to work on this project and submit it to the science fair, and we'll see how they do. The idea is that it's a proof of concept for airborne eDNA using the NYBG forest. Now the forest is not huge, but it's big enough to do something like this. And it is very well characterized in terms of how many species there are and what they are. And therefore, it's a perfect kind of testing ground for this. If this works, it will be a much less labor-intense way than doing vegetation plots, which not only take a lot of work, but also disturb the forest because you have to kind of get to every plant. In case you're curious, it's basically a small sterile filter, which the air passes through, and we capture it, capture the DNA, and then we use a universal mini barcode to amplify and then sequence and quantify. Here's the sampling device at the top of the press end. You can see the filter right here, and this is the pump, which is pumping the air through. About 50 liters of air sampled, and we will know in January whether this worked or not. Switching slightly to biodiversity informatics, the specimens that you collect in a survey are sort of the raw materials for further informatics. This involves writing a lot of code, and at the moment I'm working on a project called Monographia, which basically is a website. And this website is meant to be something like Google Docs, but for biodiversity data. The idea being that one can import specimen data, measurements, literature citations, and combine them all together into all of the things that we normally need to produce, descriptions, keys, etc. The idea is also to allow us to work better and faster, and I will talk about some of that in a moment. The basic unit of information in Monographia is a specimen. And one specimen and its measurements don't really amount to much. But as one samples additional specimens, eventually one can see trends, and those trends coalesce into patterns. Those coalesced patterns tell us a lot about the natural world. In this case, the geographic distribution of Juniperus communis in northern North America. Monographia is designed to output not only graphs of individual morphological characters and as they relate to environmental gradients, but also descriptions at a variety of reading levels. So from the professional to the field guide, and in different languages simultaneously. And as one adds more data, all of these outputs are updated automatically, so one can continuously improve a flora or a checklist as needed. Now, as I said, we get a lot of measurements from specimen. We can sequence the DNA, We can look at them under the microscope and look at fine detail. We can take individual measurements of particular parts, and we can orient the specimen in space and time. If we're lucky, the specimen will tell us exactly where it's from in terms of latitude and longitude, geographic datum, as well as time. Not all specimens are like that. Before handheld GPS was a thing, many people collected and gave landmarks, such as this specimen, which tells us exactly where it is. It's a railroad station in Mount McKinley National Park at mile 0. So it's not very hard to find on a map, perhaps. So all you really have to do is look up in a database such as GeoNames or OpenStreetMap. But this is a little tricky because the specimen is more than 50 years old, and Mount McKinley is now called Denali. The railroad station actually has moved since 1964, so you need the historic one, not the current one. And this will take you a bit of time and local knowledge and or research to figure out exactly where the specimen was collected from. So I'd like to automate this a bit so that we can take this text and simply get the latitude and longitude so that we can do further analyses. So I'm currently training a neural network in collaboration with a student at NYU, Tyler Clabby. And we're taking the data from GeoNames and OpenStreetMaps and combining that with the data from the specimens that have explicit latitude and longitude on the labels, passing them through the neural network to train it to recognize where that particular place is. So in the future, we will be able to input this text and get out an exact locality with an error distribution. Another feature that I'm trying to add to Monographia is called Grimoire. Now, Grimoire as a word is a personal spell book, which sort of has mystical connotations. And the idea of this is it's to greatly increase our ability to work. So normally you take a level apple specimen like this and you measure some things like height and width, and then you have to measure some more. The idea is that instead of measuring more, you feed this into the Grimoire model, and we retrain the model on just a little bit of data like those measurements. And now we have a new model, which is trained to recognize this particular morphological feature and tell you how big it is. So now when you feed a few measurements in, it will give you a whole bunch of measurements out. And the user can then check each one of these measurements to make sure it's okay. All of the approved ones can go back to improving the model. The personalization part of this is that essentially for every small group of plants and every morphological character, there has to be a separate model. But fortunately, it's a small model, so it's easy to retrain and build and improve upon. Lastly, I want to tell you about a aspect of Monographia that if you use it, you will probably use all the time and never even realize it. Now, we as scientists have kind of gotten good at speed reading and can skim this text and pull out all of the scientific names so that we can, link them to other things and use them as search terms. It's useful for a computer to be able to do this too, so that it can give you, for instance, descriptions. It can find morphological data in text, and it can search using the taxonomic hierarchy, not just the name that it found. So it's rather easy to do this, as it turns out, if you use 3 neural networks in combination. Now, these 3 neural networks individually don't perform that well. However, when you combine them, they perform quite well. The idea of this curve is that it's to show you the area under the curve, in this case, precision and recall. Ideally, the curve will kind of go from 1 all the way across and down. Doesn't quite do that, but it gets close. It's in the upper 80s. And with a little improvement, it will probably be in the 90s to 100. I'm going to switch now to talk about diagnostics a bit. And this involves everything from herbarium specimens to DNA sequences. And there are a lot of collaborators involved in this, both for the DNA sequencing part of it and also for the imaging. So first, the imaging. Most of us as botanists are used to and actually really enjoy determining what species a plant is. And the idea of this is essentially to teach a a machine learning algorithm to do the same so that we can be left with the interesting specimens rather than the boring common things. We have been sponsoring annual Kaggle competitions with data sets not only from our own institution, but other institutions. And the idea of this is to stimulate innovation. Data sets made available to the machine learning community result in all sorts of innovations. That's how we got, for instance, the ImageNet classifier. Someone made a data set and set it out there. And so we're doing the same with the hopes for kind of biological specific innovations. The ultimate end goal of this is to make a tool called iCurate, which is an easy to use application that can access these Kaggle models without having a machine learning background. So there's a web prototype available now. You simply drag or upload an image and it runs it through the neural network for you and gives you a prediction as to what it is. Now, machine learning by design, unfortunately, has to give you an answer. So you give it an image, it will give you an answer. So if you upload an image of this to this, of say a dog, even though it's trained on plants, it will give you a plant name, obviously the wrong name. So I'm developing a neural network architecture called Novum. The idea is that I would like the neural network to tell you when it doesn't know. And it may tell you that the species is unknown, and that could be because the neural network has never heard of it, or it could be because it's genuinely new to science. And the output essentially will tell you, for instance, that it's an unknown species in a known genus, i.e. it will tell you what it does know and then what it doesn't. And this will help us do our jobs much faster and also hopefully find new species that have already been collected and are sitting in the herbarium and just need to be described. To prepare these data sets is quite a task. We're talking data sets of 1 to 2 million images, so quite a lot of records. So we extract the specimen records from GBIF and iDigBio, and then we standardize the taxonomy after deduplicating all of the records. And there's, of course, taxonomic issues as well as data input issues that need to be solved in standardizing this. And we've gotten it down so it's relatively fast and easy. Last year, we standardized to the Leipzig list, and this year we're standardizing to the Q's World Checklist of Vascular Plants. Now, once we have a list of records that are standardized in one taxonomy, we can match those to a checklist for a particular geographic region and download all the images, which again doesn't sound like a big task, but at 2 million downloads, that's quite a lot. We also have to clean the image data because there are a fair number of records that are, say, of a living plant rather than a herbarium specimen or a close-up of a label or something like that, which we need to filter out. And lastly, we have to resize and blur the images. So resize them so that they're of uniform size because neural networks cannot tolerate missing data or, varying sized images very well. Blur the labels because, of course, what we're asking the neural network to do is to identify the plant to species. And if it does identify it to species, it's rather easy because it's written on the label typically. And we don't want the neural network to learn to read. So if you look to the side, you can see above an image, an input image, and below you can see that same image with the specimen label obscured so that the neural network focuses on the plant and not the label. This is from last year's competition, and this is quite a chart to understand. So there are about 20,000 dots here, a bit more. And this is all of the errors that the top neural network in the competition had last year. Not the good things, we don't care about those, we want the errors. Across the x-axis, you can see the phylogenetic position. So just imagine a tree down here. We used a particular tree that's published that has been time calibrated. So all of our branch lengths are measured in millions of years. But you can see the most common orders here in the data set. And then you can see every single specimen image that was misidentified. Now, an important thing is this band highlighted by arrows. This is the median distance between sister genera on this tree, which is kind of the magnitude of mistakes that people make with a 10 to 90 quartile range, sorry, percentile range. So below that band is essentially the kinds of mistakes people make, misidentifying things to the wrong species in the right genus, or perhaps the wrong genus, but a closely related wrong one. And above that band are essentially the mistakes that are rarely made, if ever, by people. It's, kind of wrong to division kind of mistakes, completely wrong. And you can see that the AI actually has output two kinds of errors. The first one is there are quite a lot of specimens that are misidentified in ways that people would never do, which is quite interesting. Although you'll notice that the distribution of dots across the x-axis is more or less uniform, suggesting that it's not any particular plant that it has trouble with, which may be surprising because you would think something like, say, Poales, that has very tiny morphological features that don't even really show up in the size of images that the neural network is looking at, would be more misidentified than something that has larger characters that are more easily seen, but that doesn't seem to be the case. The other kind of error it outputs are exactly the kind of errors people make. So, the wrong species in the right genus kind of thing. And it's unclear whether this is the neural network actual errors, or if these are errors that the neural network is imitating, because it's very possible that there are mislabeled specimens. In fact, there certainly are mislabeled specimens in this data set. And because of that, it may be imitating the kinds of errors people make rather than actually having its own errors like people. It's not clear. Another thing we learned from this competition, these are the top 5 models entered into the competition, is the effect of training images, so the number of specimens that each neural network saw. And as you can see at this point, which is 10 images per taxon, the error rate sort of starts to stabilize. Below that, it's quite low. We had as few as 3 images per taxon. And in our future competitions, we're not going to repeat this error, which is to say that we'll have a minimum of 10, which seems to be quite good in terms of its error range and its stability. Once you get up to 100, the accuracy sort of peaks at about 90%. There are some neural networks that do better with more images, which is more or less what you'd expect. And there are other neural networks that do much worse, which is an interesting effect. Perhaps that will be ironed out with additional training, perhaps not. But for the upcoming competition, we will limit everything to between 10 and 100 images, hopefully maximizing the quality of the neural networks that come out of this effort. I'm now going to switch from specimen images to DNA. One of the kind of identification tools I've worked a lot with is DNA barcoding. This is just one small example that was recently published from one of my students, a master's student from NYU. And this is devil's claw, which you can see in the picture on the side that has these sort of fantastically hooked fruits. There are only 2 species of it. Harpocophyton procumbens and Harpocophyton zahiri, and they're both from Southern Africa. This is an herbal supplement that is primarily taken for pain relief, and there are a number of clinical trials that show that it is an effective pain reliever. It is the below ground parts, by the way, that are consumed for that, not the hooked fruits. Now, due to kind of historical accident, only Harpocophyton procumbens can be sold in the United States as an herbal supplement. And that's an important distinction because in the European Union, both species can be sold. We used a PSBA-TRNH mini barcode to examine this particular species and set of herbal supplements. We assembled a reference database and examined some supplements. This is the entire mini barcode. It's not very large. And the reason for this is that the processing of plant materials to be included in herbal supplements does quite a number on the DNA. The quality is extremely low, so you have to focus on small pieces if you want a reliable assay. I've summarized these sequences by taking out all the invariant bases, so just focusing on those The two species are highlighted, and you'll see that there are three positions in this short little stretch that unambiguously distinguish the two species in Harpagophyton from everything else in the family Pedaliaceae, the sesame family. In addition, there are two positions that distinguish the species from each other. And you'll quickly realize that every one of the supplements that we sampled, bought in the United States, labeled for United States sale, is actually incorrectly labeled because they all contain Harpagophyton zahiri, which is cannot legally be sold in the US. The interesting part about this is some of them are mixtures of the two species, and you can see that quite clearly in the Sanger chromatograms, that at every diagnostic position, there is a mixture of peaks suggesting that we have both species present in this particular sample. I'm now going to shift over to talking about phylogenomics. And my focus on phylogenomics is really to make it more efficient and better. And this work is in collaboration with researchers in Gloria Caruzzi's lab at NYU, as well as Michael Tessler, who is at St. Francis. Now, you're probably all used to phylogenetics, and phylogenomics is sort of phylogenetics, but bigger. So for phylogenetics, generally speaking, you generate or download some sequence data, maybe a bit of both. You align the sequences for each locus, and you then reconstruct the trees, usually as sort of an error check, and finally combine them together to make one total evidence tree and calculate some sort of node support or tree support. And typically you conduct some sort of comparative analysis as well, but that's not required. It depends on what kind of study you're doing and what you're interested in the particular data set. Phylogenomics is very similar, and you start in the same place. You just generate a lot more sequences and or download more. Unfortunately, because of the way it works, you have to assemble the sequences as well. And for most phylogenomic pipelines, you need to extract coding regions. Once you have extracted the coding regions, you have to determine orthology. And when you are doing phylogenetics, typically this isn't a problem. You knew more or less what it was before and only have a little bit of kind of detail to work out rather than major realignment, so to speak. You then align each of the orthologs separately and construct trees for each ortholog set. You then have to filter out paralogs and also whole data sets in some cases. Some loci just don't seem to be behaving themselves, have too much missing data, or have other issues that you need to filter out before you make the total evidence tree. And this is particularly a problem in the sense that these trees take an incredibly long time to compute. So you want to do a very good job of pre-filtering before you actually go to the compute step. And finally, you actually calculate node support or tree support, and typically you also conduct some comparative analysis. So you can see in orange, we've added quite a lot of steps, and each one of them is not just for one locus, but it's for thousands, like 20,000. Most of this work has been on a pipeline that has its home in Gloria Caruzzi's lab called Phylogenius, and it ingests whole genomes or transcriptomes, so it doesn't do the assembly, but it takes the assembled data and then does gene clustering, in this case using Diamond and then MCL, and then makes alignments for each of the clusters, and then a gene tree from each of those alignments. That gene tree then has to be processed by ortholog ID to figure out what orthologs are. So you break things up into individual loci at this point. And then we have to concatenate the orthologs into a total evidence analysis. And that total evidence tree, as I said, takes forever to run. And eventually we have a tree that we can then talk about and compute support statistics for and do other kinds of comparative analyses. Now, this pipeline, even on a modest data set, takes quite a long time to run on a high-powered computer cluster. We've spent a lot of time optimizing it for fast parallel processing. refining the algorithms, making heuristics to do less on the larger parts of the data set, etc. But it still is quite intense. And we need to improve it in some way. So one way of doing this, of course, is replace existing algorithms with either a new algorithm or a shortcut to that algorithm. And one kind of low-hanging fruit is the fact that in the process of this pipeline, we compute trees many times, more than I actually showed you in that last diagram, but we don't actually use all of those trees. A lot of times we only use some score from the tree, for instance, the length of a parsimony tree or the likelihood score from a tree. And so we don't necessarily need the tree, we just need the value from it. And since this is used quite a lot, these trees, it would be nice to replace at least some of them. Now, of course, the problem with this is new algorithms are hard to make. Anyone who has tried to dream up an algorithm often ends in frustration. And this may be something that machine learning can help us with, but we haven't tried applying it yet. So the first question we asked is, can we take existing algorithms and repurpose them for our process? So to figure out whether we can do that, we first assembled a list of existing algorithms, 21 in fact, and we assembled some phylogenomic data sets. One is from Brassicaceae, another is from Drosophila, and a third is from fungi. These were selected because they're very well characterized large data sets for phylogenomics, and they're publicly available. So in the end, after extracting, processing them, and extracting all the single copy loci from each one of them, we end up with 20,000 loci to be sort of our test data set. Now, these data sets are relatively small in terms of the number of terminals. They're between 10 and 30 per locus, so not particularly large in that sense, but there are a lot of them. And so we added a fourth data set, which is bacteria. Now, bacteria have the nice property that you can fully sequence their genome relatively easily. So we assembled a data set of 1,000 terminals and 30 loci without any missing data. And the reason for this is this will allow us to test scaling a bit, which we can't test with the phylogenomic data sets. So first, the kind of phylogenomic in terms of loci, we ran all of these analyses and then looked at the clustering effect. And so we did two different clustering techniques. The lower one, the scatter diagram, I think is actually more useful, although both of them come to the same general conclusion. And the only things that would be surprising with these metrics really is where certain things went. So likelihood score, for instance, was most closely related to the parsimony consistency and retention index rather than the other likelihood values, such as the likelihood support or the sum of the likelihood support, which also went in an interesting place with kind of matrix descriptors. Now, this diagram is also a bit difficult to read. In blue, it shows the correlation between individual algorithms, with the darker color and larger dots indicating a stronger correlation, and the lighter or no dot indicating no correlation. The bottom in shades of red show the time relationships, and I'll get to that in a moment. So let's just focus on the blue to begin with. So first of all, the thing that is probably obvious but nice to see when you run an analysis that takes this long is that the different tree building metrics are more or less correlated with each other, which is to say the likelihood trees and the parsimony trees more or less look the same. They usually do in most data sets, but here are 20,000 data sets that show that they almost always do. In addition, one of the interesting things that comes out of this is that there are a lot of values in the parsimony realm that are highly correlated. So the parsimony tree length, which takes a long time to calculate, is almost exactly correlated with the parsimony information and its two components, minimum steps and maximum steps. In addition, a lot of shall we say, matrix statistics are correlated with that, such as the edit distance and the sum of pairs, which is also correlated with the likelihood scores. conveniently. And lastly, I just want to point out, although this is probably not a surprise to most people, that no matter how you calculate sum of pairs, no matter which matrix you use, they basically all come out to be the same value. Not surprising, but important since people fight about which matrix is better. In terms of time, the thing that most dominates the time plot is the likelihood support values, which take for absolute ever, which is not perhaps a surprise to anyone who's ever computed them, but there you see it, averaged over 20,000, it takes forever. The other thing that dominates this plot is something called TCS, Transitive Consistency Score, which is an alignment rating that you've probably never used and probably never will use, and I wouldn't recommend using it given how slow it is. To test the scaling, we looked at the 30 bacterial loci, and so we took our 1,000 terminals and subset it from 75 to 1,000, so you can kind of see the scaling effect. Now, some algorithms, most of the ones that we tested, in fact, have near linear scaling, which is to say instant, even on 1,000 terminal data set, which is very nice because these are algorithms that, if correlated with something else, will be incredibly useful because they already scale well. Now, if you look at kind of the poster child for something that doesn't scale well, the maximum likelihood tree score at 75 is kind of reasonable in terms of the amount of time it 75 terminals is reasonable the amount of time it takes to compute. But as soon as you get up to a reasonable amount of terminals, in my opinion, 1,000 is not a huge data set when it comes down to the diversity of life. It takes forever. And of course, you can see this is near exponential scaling, which means that it's only going to get worse. And this is definitely an algorithm which we need to replace with something faster if we want phylogenomics to work at scale. So from these results, we can predict that one can save not only hours, but days, weeks, and months, as well as some money. So for a hypothetical data set of 20,000 loci, which is kind of typical for a phylogenomics scale data set, and 1,000 taxa, which is much larger than most of them are currently, although strangely enough, one can go onto GenBank and get 1,000 plant transcriptomes or 1,000 animal transcriptomes. So the only thing that's really stopping people from doing these analyses is the computer power, not the data. So if we take locus filtration, which is one tiny step in the pipeline that I showed you a few slides ago, you'll see that the minimum parsimony steps, for example, is 87% correlated with the likelihood support value, but the time to compute that is 7.3 hours for 1,000 taxa and 20,000 loci versus 17.6 years. Of course, that's on one processor. No one uses one processor. We use computer clusters with many processors. But the point being, if you had to pay for this directly by, say, renting a cloud service like Amazon, it would take you about $2 worth of compute time to compute minimum parsimony steps for this data set. And $32,000 to compute the likelihood support value. So you can see the monetary and time savings is immense. Now I should point out, we didn't actually optimize things like minimum parsimony steps. We just sort of implemented them in the quickest way to program. And the reason for that is we knew they would be fast, and spending a lot of time on optimizing something that's not necessarily going to be useful didn't seem like time well spent. But now we see that how useful they are, we can probably optimize them much further and get that time down to maybe an hour. The important thing about this is this saves vast amounts of time. It also saves a lot of money, especially if you're renting computer time, and a lot of carbon emissions, because you can imagine $32,000 worth of compute time comes with quite the environmental cost. So I'd like to thank you for your attention and just sort of summarize that I've told you a bit about biodiversity inventories, biodiversity informatics, some diagnostics, and some phylogenomics. And it may seem like sitting in front of a computer and writing codes or training a neural network is far from what we kind of signed up for in terms of expeditions and collecting plants in the field. But I would like to think that this work actually allows us to get things done faster and better so we can actually spend more time in the field and, more importantly, do our jobs more efficiently than ever before. With that, I'm happy to take questions as they may arise.
Okay, Damon, thanks very much. What a fascinating presentation. I'm going to read some of the questions from the list of questions people have submitted. Maybe I'll start with a real easy one first
Sure.
from one of our viewers. Was Fisher and machine learning, sorry, scrolling around here. It's the same as the Fisher's exact test in statistics.
Yes, it is. Ronald Fisher was quite the, we'll say, mathematical and statistical genius. So he worked both on early machine learning, various kinds of statistics, population genetics, a wide variety of things.
All right. We have another viewer who asks, I've read a lot about the use of LiDAR for archaeology. Would LiDAR images of forests provide useful data for the type of work that you're doing?
Oh, absolutely. LiDAR is at least been proposed to be used for this kind of thing, from either space-based from the International Space Station or from aircraft. Mostly the reason why we haven't included it is that LiDAR data are sort of spotty in the sense that they historically don't go back very far and also don't have global coverage. And I'd like to make this kind of as generally useful as possible, both for historic changes in biodiversity, but also to cover the entire planet if possible.
Okay. We have another viewer who asks, would knowledge of computer science be essential for future botanists? Should a budding botanist learn how to code?
That's a very good question. I think that we've gotten to a point now where I would say most botanists are in that vein. If it's not, we'll say, serious, kind of optimizing kind of level coding, but certainly, using, say, R or Python to analyze large data sets, calculate things that can really only be calculated using code, even if it's not, kind of the advanced optimized kind. So absolutely, I think it's a very important skill for analyzing data and for publishing papers in a relatively expedient way.
I'd agree. I think the education students are getting now is quite different from the one that some of us old timers got. Somebody asks, is machine learning currently used to identify plants of potential medicinal value? If so, where and who might be working on it?
That's a good question. I haven't seen anything about that, although I would think that it would be kind of a ripe topic. It might be worth spending a little time looking in the machine learning literature. It is a very difficult literature to search in the sense that people who study machine learning often use terms that biologists don't and vice versa. So you kind of have to learn two languages, so to speak, in order to search it, but it would definitely be worth doing because I imagine someone is, if not working on this now, will be soon.
All right, and we have lots of questions coming in, but I think we only have time for one more. So let me finish with the, it looks like the most highly rated question. Maybe I misunderstood about the NYBG barcode project, but how do you get plant DNA by sampling the air?
Oh, that is a good question. So there have been a number of studies using dust traps, for instance, to sample airborne DNA. And they've conclusively demonstrated that one can get DNA from plants in the local area that is not from shed pollen, because both time and abundances were sort of off for that. So usually what one is getting is actually bits of plant. So there are hairs, for instance, on a lot of plants that break off, or little bits of leaf, bits of bark. And so you're basically getting plant dust in the traps, and those contain enough DNA remarkably to be sequenced.
All right, well, Damon, you and me, we need to talk a lot about some interesting collaborations, and this has really been fascinating, and the question's been great, and I really appreciate, we had a very good turnout today. Appreciate all your time and everybody's attention, and but it's time to wrap up today's webinar. If you missed any part of it, or you would like to watch it again, a recording has been saved and will be published in the NYBG lecture library. And we also invite you to please join us for future science webinars. The next one coming up is later this week on Thursday, December 23rd, to be held at 10 a.m., which is a presentation by Dr. Barbara Ambrose called Building Beauty Patterns in Plant Biodiversity. Those of you who have not already registered may sign up using a link that'll be posted in the chat in a few seconds. And until next time, please stay well, stay green, and take care. Goodbye, everybody. And thanks once again, Damon, for an amazing presentation.
Thank you very much.
All right, take care. Bye-bye.